Hey guys, let me just check if the stream is live. Okay, great. So go and hit the like button before we start the video because today we're going to cover something that is very important for your prof examinations. It's slightly more important for your prof examinations rather than uh, for your NEET PG examination. But so we are going to cover gene regulation in prokaryotes as well as that in eukaryotes. So I'll tell you which specific topics are more important for NEET PG as compared to which are more important for your prof examinations. As a whole, this entire topic is very important for your prof examinations, and the portion of prokaryotes is relatively more important for your NEET PG, and some part in the eukaryotic portion is also important for your NEET PG. So I'll tell you at the relevant places where what you have to do. Before that, let me welcome you to Unacademy and Let's Crack NEET PG, the YouTube channel for the Unacademy's NEET PG division. So you can join us on Telegram. The Telegram channel link is t.me/Let's Crack NEET PG. So you can join this Telegram channel for updates regarding what goes on this channel, which educator is taking what course, right? So about me, my name is Chetanya Mittal. I am an MBBS from All India Institute of Medical Sciences, that is AIIMS, New Delhi, All India Rank 4, and JIPMER, All India Rank 5, right? So today we are going to be discussing regulation of gene expression in prokaryotes and eukaryotes, alright? You might have read this chapter in your class 11 and class 12, but I swear this is in much more detail as compared to what you would have read earlier, right? So before I begin, I must tell you that from where you should read this chapter. I would really recommend that you read this chapter from Lippincott. But even if you refer to my lecture notes from this class, they are going to be sufficient for your any entrance examination or for your any examination that you appear in. They're going to be pretty sufficient, right? So before I begin, I would request you all to download the Unacademy Learning app. Go to the Play Store, click on install. It's very simple. And put in your phone number, put in your ID and everything. And all you have to do is go and purchase a free plan for NEET PG, right? So there is a free plan option. For that, you will need my referral code, and that referral code is Chetanya10, right? This is the referral code that works to purchase a free plan. If there is anybody who you know who is unable to install Unacademy, do tell them about this referral code, okay? So these are all the educators who are teaching on Unacademy. You can see they are the top educators for their respective subjects, and you can see some of them are also from my college. Special classes on Unacademy are the free live classes, which will give you an experience of what Unacademy Plus is like. So you can go to my profile. This is my profile link on Unacademy. You will also find it in the description box, and these are the things that you can see here. You can dedicate me a hat if you like my lectures. You have the option of dedicating a hat, and so these are all the special classes that I have made for you guys, and you can go and check them out if you are interested in watching any of these topics. Besides that, there are plus courses that are made by top educators, and I have also made a plus course for you on GI physiology, right? And if you are interested to know about it, you can go ahead and check it out. There's the details of the course are there in the course description, right? And then you have running batch courses for syllabus completion. INI-CET batch starts in January. So if you want to take a subscription, now is the best time to take a subscription, and do use my referral code when you take a subscription, right? Then there are free mock tests that you can attempt on the app, and then you can see that there are multiple mock tests that keep appearing, and you can attempt these as many times as you want. Unacademy is the easiest way to learn online in terms of flexibility, whether it be for time, whether it be for language, or for unlimited practice, or unlimited times you can watch this video, unlimited times you can rewind and come back. There's a very robust chat feature here also on YouTube. I have opened the live chat on my mobile phone. So whatever you guys write, I can see here. So please do make the class interactive, do ask your doubts. And I have some MCQs at the end, so we'll do this as interactive polls, and you have the ability to learn anytime, anywhere you want. So go ahead and install the Unacademy learning app. You'll get the best of Unacademy and Prep Ladder if you take the iconic subscription, where you not only get the live classes of Unacademy, but you also get all the videos, the question banks, and the rapid revision courses of Prep Ladder, right? So these are the differences between Unacademy Plus and Iconic. There are some additional features of Prep Ladder that you get here, and I would recommend that you go with the Iconic subscription for either the 1 year or the 2 year. The 1 year subscription costs 55,000 and the 2 year subscription is 77,000. So I would say that go with the 2 year subscription and apply my referral code Chetanya10. You will get a straight 7,700 rupees discount. ठीक है? तो 8,000 का discount छोटी बात नहीं होती यार. So just use this referral code and take this discount, right? And you will also be able to apply credits and everything. ठीक है? And you have the option of no cost EMIs. ठीक है? I know 70,000 is a huge amount and it's very difficult to pay it in one go. So take the EMI plan. It's a no cost EMI. There is no harm in taking it, right? You can take it either with a debit card or a credit card. Then this is the Plus subscription. Plus subscription, again, I would recommend the 1 year and the 2 year plans. It's an Unacademy secret that these plans are priced the best, right? So go and click on apply for your applying the referral code and applying the credits. Then go ahead with your transaction. So we'll begin today's class on regulation of gene expression. So today we are going to talk about regulation of gene expression in both prokaryotes and eukaryotes. We're going to talk both about prokaryotes and eukaryotes. I hope you guys are all MBBS students or medical students, BAM students, right? Or preparing for FMGE, whatever way you're doing that. And I hope you know what are prokaryotes and what are eukaryotes. Prokaryotes are your bacteria and everything which have a false nucleus. Eukaryotes have a true nucleus. That's a very basic thing. So one important difference between prokaryotes and eukaryotes I want you to know before we begin this chapter is the fact that in prokaryotes transcription and translation occur simultaneously. ठीक है? Transcription खत्म होती नहीं कि translation शुरू हो जाती है. Why? Because here you do not have any cytoplasmic differentiation with the nucleus. ठीक है? कोई nucleus और cytoplasm अलग-अलग नहीं है यहाँ पे. So transcription, which otherwise usually occurs in the nucleus, and translation, it will go and occur outside on the ribosome. In case of the prokaryotes, there is no such differentiation. There is no such spatial separation. So here transcription and translation are coupled in time and space, right? Transcription and translation are coupled. ठीक है? Transcription start होती है, उसके कुछ seconds या minutes के बाद ही translation भी start हो जाती है. ठीक है? DNA is being converted to mRNA, and on that mRNA, the ribosome also starts moving and forming peptide. Alright, so that's one important point of difference. Whereas in eukaryotes, first the entire mRNA will undergo transcription, and first heterogeneous RNA is formed, and then that is processed to mRNA, and then finally that mRNA moves out of the nucleus and you have translation. So here it is possible to regulate both transcription and translation, whereas in case of prokaryotes, transcription is the main site of regulation. Okay, so that is one difference that you must understand between prokaryotes and eukaryotes before we begin the class, right? Is that much clear to you? You guys can say hello. If you guys have to say something, you guys can stop me anytime. You can ask your doubts. Do hit the like button on this video. I am sure you will like it. Okay, so Tushar Sharma, Aditya Mirzapure, if you guys have anything to say, you can drop a chat anytime. So should we begin the class? Should we begin the class ahead? I've given you a general introduction to what we'll be doing. What are the prerequisites before we start studying, right? And then let's begin. Right, so let's begin the class. The first thing is that in case of prokaryotes, I told you that the only regulation of gene expression that is possible is at the level of transcription. It is only possible at the level of transcription. Why? Because I told you that once transcription has started, translation is going to occur unless the transcription is terminated prematurely, right? So at the level of transcription, there are two ways to regulate transcription. One way to regulate is that you control the initiation of transcription, that when does transcription start and when does transcription not start. So this is a on and off switch. It is like a on and off switch, right? So you can control whether you have to turn the switch on or whether you have to keep it off, right? So that's one thing for initiation of transcription. The second way of regulating it is premature termination of transcription. Okay, I'll tell you what this is. This is basically called as attenuation. This is basically called as attenuation by a transcription termination loop. Okay, abhi I'm not going to talk about this for now. Let's focus on initiation of transcription. And here you know the main regulation you've studied in your class 11th also is done via operon. So we are going to study two types of operon, the Lac Operon and the Trp Operon. This you might have studied in detail in class 12, right? But here also you did not study the catabolite activator protein. You did not study the catabolite activator protein. Trp Operon, you know, is going to have two kinds of things. It is having the regulation of initiation of transcription, and here you will also see the regulation that I told you about, that is called as attenuation. Yeah, so I'll come back to attenuation. Let's first start with Lac Operon. So in Lac Operon, the first thing is that there is a promoter, there is an operator, there is three genes that are present together because they have to be regulated together: Lac Z, Lac Y, and Lac A. One of the common questions that used to be there was Lac Z is coding beta galactosidase, Lac Y is coding permease, and Lac A is coding trans acetylase. It is coding trans acetylase, and they are involved in lactose metabolism. Alright. So then there is an operator site. And if a repressor is bound to the operator, if a repressor is bound here, then that repressor is going to stop the RNA polymerase from moving forward. That is going to block the RNA polymerase from moving forward. So therefore, if the repressor is bound here, that blocks the RNA polymerase is given here, right? And the second thing is that there is a promoter site here where the RNA polymerase actually goes and binds before it starts to move on the DNA. And then there is a cap site. What is this? Catabolite activator protein site, right? So here what happens, what binds here is the protein that binds here is known as CAP, that is catabolite activator protein. It is a protein that comes and binds here, and this protein, it will promote the binding of RNA polymerase, right? So this is positive regulation and this is negative regulation, right? So remember in your NCRTs, it was written that Lac Operon is also under positive regulation, but that is beyond the scope of your syllabus. So this was the positive regulation that was being talked about. In your NCRTs. कुछ चमका दिमाग में? ठीक है, I'll tell you how this works. I'll tell you how exactly this works. But for now, know there is what are the components of the Lac Operon: promoter, cap site, operator, Lac Z, Lac Y, and Lac A. There is a gene here that is known as Lac I, which synthesizes repressor. It synthesizes repressor, and this gene is constitutively active. What do I mean by constitutively active? It means that it is functional all the time. That is, it is synthesizing its product all the time. It is synthesizing the repressor all the time. Alright. So Lac I gene is constitutively active. It is all the time synthesizing the repressor, which goes and binds here to the operator site. Okay, and blocks the movement of RNA polymerase. Do you understand this so far? Cool. Shah Sharma has written a yes. So the first thing that is going to be there is that when there is low glucose and lactose is available. So let me show you here what happens when there is high glucose. When there is high glucose, there is high amount of this high amount of inhibition of this enzyme called as adenylate cyclase. See, if there is high glucose, this adenylate cyclase enzyme is inhibited, and if this enzyme is inhibited, there is less production of CAMP, right? And if CAMP is inhibited, if CAMP is not being formed, then catabolite activator protein, it does not bind here to the site, that is the cap site, and if it does not bind to the cap site, there is no positive feedback for the RNA polymerase to bind here. So there is no transcription. That's the first thing. The first thing that you have to understand is for glucose, and the second thing that you have to understand is for lactose. When there is high levels of lactose, that is present here, these are the lactose molecules, they will go and bind with the repressor. So lactose molecule, or rather allolactose, it acts as an inducer. For the Lac Operon. Why? Because inducer goes and binds with the repressor, so that the repressor cannot go and bind with the operator, right? So now once there is high amount of lactose, if there is high amount of lactose, then what will happen? The repressor is inactivated, and the operator site, it is free. Operator site is free. So transcription can occur. RNA polymerase can easily move over this. There is no inhibition of RNA polymerase. So that's the first thing that you understand, and this you have also studied in your class 12. What you have not studied in your class 12, what you will study in your first year MBBS, is the fact that when there is high level of glucose, that will inhibit adenylate cyclase. If adenylate cyclase is inhibited, then ATP cannot be converted into CAMP, and CAMP is required for the binding of catabolite activator protein to the cap site, right? And if it does not bind here, then the RNA polymerase cannot come and attach to the promoter site. Why? Because I just showed you the cap site promotes the binding of RNA polymerase. So the cap site must be occupied and the operator site must be empty for binding of RNA polymerase. And you've just seen how the cap site is occupied. So if I reverse this, what is shown here, if I say there is low level of glucose, right? So what will happen? That will activate the adenylate cyclase, that will cause conversion of ATP to AMP, and conversion of ATP to AMP will enhance the binding of the cap and the cap. They will join together, and that will go and bind to the cap site. So it's all a game of the CAMP levels. Camp levels, they will regulate the binding here, right? You get it? So now let's see 4 cases. If there is low glucose and there is availability of lactose, because there is low glucose, cap site is occupied, and because lactose is available, the operator site is unoccupied. Okay, so that will favor transcription. Cap protein and cap complex are present here. Cap complex, they are present here. They will favor the binding of RNA polymerase, and the Lac genes are strongly expressed. In all other cases, there is very little or basal expression of Lac genes, right? See here, if there is high glucose and lactose is unavailable, then repressor protein will be bound, so there will be no transcription. If there is low glucose and again lactose is unavailable, then again this situation will be there where there is repressor protein and there is no expression of Lac genes. Only when there is availability of lactose, there will be binding of glucose to the, only then there will be absence of repressor, operator site will be free, and only then can the polymerase function. In that, if there is high glucose, then the cap binding site, that is going to be unoccupied. So there will be very less transcription. There will be very less or basal amount of transcription. Okay. Only when the cap site becomes full, with the help of CAMP, only then will the Lac genes be strongly expressed. So that is the regulation of transcription that I wanted to explain with the help of Lac Operon. Have you been able to understand this? What happens in the absence of lactose? Right? In the absence of lactose, repressor is activated, it goes and binds to the operator, and there is no transcription. If the lactose is present, the allolactose, it binds here with the repressor, and then there is transcription and translation of the three enzymes. There is transcription and translation of the three enzymes, right? So I hope you guys understand this entire cycle. I hope you guys understand this entire cycle. You guys are okay with all of this? Is everything clear for you guys? Yes or no? Come on guys. Yes or no? Yes or no तो लिख दो, और कुछ नहीं लिख रहे तो. Tejas Nandania is saying nice explanation. Great. चलो. दिल खुश हो गया ये बात सुन के. So should we move to TRP Operon? Should we move to TRP Operon? Next we'll move to TRP Operon. Okay. So should we do an MCQ before we move ahead? Let's do an MCQ before we move ahead. एक MCQ कर लेते हैं. Question number 1. Which of the following are situated away from the coding region? This is a previous year question of PGI, June 2006. So hit the like button if you like what you have been seeing so far. If you like the video, if you like my explanation, please do hit the like button, and if you can, do drop a comment later on so that people know that the video is at least worth watching once. Okay. So what is your answer for the first question, guys? You should be able to answer this. Try to answer this based on exclusion. Even if you don't know the answer, I just showed you a diagram. Try to answer this based on exclusion. Okay. So Tejas Nandania has said that the answer is A, promoter. Okay. Read the question very carefully. It says, which of the following are situated away from the coding region? Okay. Try to answer this. The operator you saw is situated just next to the coding region. Structural gene is part of the coding region. So D is wrong. Shabstazine. D is not the correct answer. Right? Humaira, hi. Right? And promoter was also very near the coding region. It was attached just adjacent to the operator region. So these three are wrong. The correct answer is option B, enhancer. Alright, when we do gene regulation in eukaryotes, I will tell you what are enhancers, right? And then you might be able to answer this a little better. Okay, so I'll come back to the theoretical part and I'll come back to TRP Operon. Let's explain TRP Operon to you. Okay, TRP Operon you might not have read during your class 11. Okay, so one of the difference between Lac Operon and TRP Operon is that TRP Operon is for synthesis and Lac Operon is for breakdown of lactose, right? So TRP Operon, the purpose of TRP Operon is to synthesize tryptophan. Okay, so when should the tryptophan operon work and when should it not work? When there is low level of tryptophan, the tryptophan operon should work, right? The TRP operon should work. And when there is high levels of TRP, then the TRP operon should stop working. This is in contrast to the Lac Operon. Lac Operon was working when lactose was in high amounts. It was not working when the lactose levels were low. So this is working via repression. This is working in a different way. That's why we are studying the two different models. Okay. So again here you will see there is a promoter site. There is an operator site. then there is something known as a leader sequence. There is something known as a leader sequence in case of Trp Operon. I'll tell you why. There is an attenuator here, and then you have the structural genes. Then you have the structural genes, and they are in the opposite order. They are in the opposite order. So first you will have TRPE, then you have TRPD, then you have TRPC, then you have TRPB, and then you have TRPA. Okay. Understand this much? Okay. Then I said that in the Trp Operon, I'll take a black screen. The regulation of transcription is in two ways. Alright. Regulation of Trp Operon is done at two ways. One is initiation of transcription. Okay. Similar to the Lac Operon. Initiation of transcription will be regulated. Initiation of transcription will be regulated. The second thing is premature termination of transcription. Premature termination of transcription. This will occur because of the attenuator. This will occur because of the attenuator. What happens? The transcription starts, but if there is high level of tryptophan, the transcription will stop prematurely. Only the leader peptide would have been synthesized. The transcription would be stopped here. The transcription would be stopped here. Only the leader peptide would have been synthesized. There will be attenuation here, and the mRNA form will be attenuated, and this will get degraded very fast. This will get degraded very fast, and translation will stop here. So there is premature termination of transcription and translation. That is one way of regulating this. Okay. Hi Sangam. Okay. So the TRPR, that is TRP repressor gene, it will synthesize a protein that is TRP repressor protein. Now the TRP repressor protein, it will be present in the inactive form. Why? Because it requires someone along with it, and what it requires is the co-repressor. What it requires is the co-repressor. Now tryptophan itself, it acts as the co-repressor. Tryptophan itself, it acts as the co-repressor. Why? Because when tryptophan is in high amounts, then it is available to act as co-repressor. If there is no tryptophan, then the repressor will remain in the inactive form. So when there is high amount of tryptophan, it will come and bind to the tryptophan repressor, and then this TRP repressor co-repressor complex will go and bind to the operator site, and it will lead to repression. It will lead to repression of transcription. So this is where you will control the initiation of transcription. Okay, very simple to understand. Nothing is complicated here, right? What is complicated here is that if there is some amount of transcription that is occurring, that is taken care of by this leader sequence and attenuation. What is leader sequence and attenuation? Let's have a look at that. If there is high level of tryptophan, right, the leader peptide, it will be synthesized, and what will happen? The stem loop here formed will give the transcription termination signal. ठीक है, तो क्या होता है basically यहाँ पे? What you have to know is that sites number 2 and 3 are complementary to each other, and 3 and 4 are also complementary to each other in the initial part, in the attenuator segment. ठीक है, so if there is high level of tryptophan, what happens here is the ribosome moves here very fast. And since it moves here very fast, the second site here, it gets occupied by the ribosome. Okay. And then the third and the fourth site, they will bind together and they will form the stem loop, that is known as transcription termination signal. If this stem loop becomes formed, even if the leader peptide is synthesized completely, this stem loop will terminate. And that will cause the formation of only this leader peptide, and that will get degraded very easily. But the structural genes, they are not synthesized. But if there is low level of tryptophan, the leader peptide is formed in an incomplete way. Why? Because for formation of leader peptide, you require lot of tryptophan. So the ribosome will stall here. Stall means the ribosome slows down. And when it slows down, the second site is available for binding with the third. So when the second and third, they will bind, that will form alternate stem loop, and then the RNA polymerase, it can move ahead and that will continue transcription. So TRP regulated genes are going to be formed. Okay, it's a little complicated to understand. Okay, I have tried to simplify it with this diagram 1 2 3 4 wala, and that's exactly how this works. It will seem very basic to you, but you need to remember about attenuation. You need to remember about this word attenuation and formation of stem loop, which is the transcription termination signal. Okay, try to remember these two words at least and try to write these down in your paper, because these are the things that will actually fetch you extra marks. Okay, TRP Operon samajh aaya? At least I don't expect you to have understood this completely, but at least you would have understood the repressor, the co-repressor, and its regulation and the logic behind its regulation. Okay, would you be able to write a 5-mark answer on Lac Operon, a 5-mark answer on TRP Operon, at least this much? Okay, because that's more or less about the main regulation in prokaryotes. That's the main regulation in prokaryotes. So if a question comes, write down the regulation of gene expression in prokaryotes. There are 4 things that you have to write. Okay, in prokaryotes, you have to write down 4 things. What are the 4 things that you have to write? First, you have to write Lac Operon. Second, you have to write the TRP Operon. Third, you have to write the R protein. I will tell you about the R protein. And fourth, you have to write down about the stringent response. I'll tell you these, the third and fourth thing. This I'll just tell you what is the third and the fourth thing that I have written here. These are the 4 things that you have to write. I've taught you 1 and 2. Let's move on to 3 and 4. Okay. So here I'm going to talk about regulation of rRNA and protein synthesis in response to environmental stress. Okay. So besides the Lac Operon and TRP Operon, there is regulation of RNA and protein synthesis in response to environmental stress. And which RNA is this? The ribosomal RNA. Ribosomal RNA. rRNA. So formation of proteins and formation of ribosomes as a whole is regulated in response to environmental stresses. And that is known as stringent response. That is known as stringent response. Stringent response, first thing that you should know is that it occurs in response to amino acid starvation. It occurs in response to amino acid starvation. That's the first thing that you should know. Stringent response, why it is occurring? In response to amino acid starvation. Okay. Then what happens? What do I mean by amino acid starvation? I hope you understand. There is an environmental stress where the cell is not getting enough amino acids. So since the cell is not getting enough amino acids, the tRNAs, they remain uncharged. What do I mean by uncharged? So if this was a tRNA molecule, right, there is no amino acid there to bind to it. There is no amino acid there to bind to it. So it is empty. So when an empty tRNA comes and attaches to the ribosome, to the A site of a assembled ribosome, that will activate a stringent factor, that will activate a protein known as the stringent factor. This is basically an enzyme. Alright, it is an enzyme which is physically associated with ribosomes. It is called as RE1A. Okay, so I remember this as ribosomal enzyme 1A because it is present physically associated with the ribosome. So it is also called as the stringent factor. Okay, so what this enzyme does is that it catalyzes the formation of guanosine tetraphosphate. Okay, you know what is guanosine? Guanosine is a nucleotide, right? And guanosine, if I attach 4 phosphates to it, so that will be guanosine tetraphosphate. GTP is guanosine triphosphate. Now I have formed guanosine tetraphosphate, and guanosine tetraphosphate is an inhibitor of ribosomal RNA synthesis. So it will inhibit the formation of rRNA, and therefore it will stop the formation of ribosomes. I hope you understand why that is happening. Why? Because cell does not have amino acids. Now cell is saying, if I don't have amino acids, why should I waste my energy on formation of ribosomes? Because ribosomes are there for protein synthesis. For protein synthesis you require amino acids. I don't have amino acids, so don't waste your energy on rRNA synthesis. So in stringent response, guanosine tetraphosphate is the mediator through which rRNA synthesis and also some tRNA and some mRNA synthesis for ribosomal proteins, these will be inhibited. Okay, so guanosine tetraphosphate, what it does is it binds the RNA polymerase and alters the promoter selection through use of different sigma factors of the polymerase. Okay, chhod do ye baat. But important thing to remember here is guanosine tetraphosphate, it takes part in stringent response. It takes part in stringent response. All right. So that's what you need to know about stringent response. See here, RE1A, I told you this is the physically associated enzyme that is known as the stringent factor, ribosomal enzyme 1A. Okay, I don't know what is the exact full form, but for memory sake, RE1A will do the job. Amino acid starvation hai ya fir antibiotic stress hai, so what will happen? There will be formation of guanosine tetraphosphate. Then what will happen? What will happen? There will be formation of something known as alarmone. It is a hypothetical hormone, alarmone, and that will cause inhibition of, that will cause RNA polymerase to mediate certain effects. It will reduce the biosynthesis of enzymes. It will reduce the biosynthesis of RNA proteins. Okay, rRNA proteins. It will form stress proteins, right? It will reduce DNA replication. Okay, so ye sare effects, they are part of the stringent response. They are all part of the stringent response. Okay. Clear? Okay. So now question, which of the following is known as stringent factor, is known as stringent factor? Choose your answers. Okay, very easy question, but just to show you how an MCQ can be framed from here. Okay, so again, you have chosen the right answer, Anthony Roy. The answer is RE1A. Very simple. I just taught you this, but just to make sure that this registers in your head that this is the stringent factor. All right. Now about R proteins, regulation through R proteins. What are ribosomal proteins? They are known as R proteins. Okay. So R protein, it binds to the SD sequence. What is the SD sequence? You know what is the SD sequence, right? It is present on the mRNA and it acts as an impediment to the binding of 30S RNA to the SD sequence. What is the SD sequence? Tell me, guys. Have you heard of this? Have you heard of this sequence? Shine-Dalgarno sequence, right? So that's the SD sequence here. So R protein, it goes and binds to the SD sequence on mRNA and it acts as an impediment to the binding of 30S RNA, that is the ribosomal subunit, small ribosomal subunit, right? No, no. Guanosine tetraphosphate is not the Shine-Dalgarno sequence. Okay, okay. You were giving the answer for RE1A, right? See, correct, correct. R protein, it binds to the Shine-Dalgarno sequence on mRNA and it prevents the binding of the small ribosomal subunit to the sequence, right? The small ribosomal subunit. So in bacteria, how does the ribosome recognize mRNA and the site at which it has to go and bind? That is done by the Shine-Dalgarno sequence. Thus, if there is R protein, thus if the R protein is present on the Shine-Dalgarno sequence, it inhibits the synthesis of all the remaining ribosomal proteins of the operon. So this R protein, it will also bind to the ribosomal rRNA and with a higher affinity than for mRNA. So if the concentration of rRNA concentration falls, then the R protein is also available to bind to its own mRNA and inhibits its translation, right? So that's what you have to write when you write about R proteins. Just remember how R proteins maintain a balance between R proteins and R RNA so that ribosomes can be formed efficiently. Okay. So now you're clear with what you have to write when it comes to writing about gene expression of sorry very basic answer, regulation of gene expression in prokaryotes. Okay. Specifically the Lac Operon and the Trp Operon part, it's important for your NEET PG. Okay. You have to write 4 things. If the question comes, you write the Lac Operon, you write the Trp Operon, you will write about the stringent response, and you will write about the R protein regulation. Okay. So again, like I told you, the best place to read this is from Lippincott. You can also read it from Harper. The gold standard place to read it from Leninger, but I don't think you guys will have this much time to read it. So Lippincott is the best book to read this from. Okay. But my notes are very sufficient for your prof examinations and even for your PG entrances. There's nothing that will remain uncovered here for any topic. Okay. Should we move on to eukaryotes? Have you understood prokaryotes completely? Only then I will move on to eukaryotes. Only then I will move on to eukaryotes. Should we move ahead, guys? You guys want a 5-minute break between prokaryotes and eukaryotes? Okay. I'll take a 2-minute break. I'm back. If you have any doubts so far from the portion related to prokaryotes, you guys can let me know. Doubts? Okay. Since there are none, let me move ahead. Right. So the thing about eukaryotes, like I told you, that the regulation is possible at multiple levels. The regulation is possible at multiple levels. It's possible at transcriptional level, it's possible at the post-transcriptional level, then it's possible at the translational level, then it's possible at the level of DNA. Okay? So there are multiple sites at where eukaryotes can regulate the expression of their own genes. But the most important point and the most important difference that you can remember is that there is nothing like operons in the eukaryote. There is something similar, but there is nothing like operons in eukaryotes. Operons is a concept that is exclusive to prokaryotes. That is there for prokaryotes. Okay, that's the first thing that you can remember. Next, when we move on to eukaryotes, the main regulation here is through cis-acting and trans-acting factors. Now, this is known as coordinate regulation. The first thing that you will write is, first point that you will make is regarding regulation at the transcriptional level. The first level of regulation that you will specify is the regulation at transcriptional level. Okay. At the transcriptional level, we will say that there is coordinate regulation. The first point is coordinate regulation. So there is a trans-acting protein which functions as a specific transcription factor. Now there are GTFs and there are STFs. GTFs are general transcription factors and STFs are specific transcription factors. They will bind to a cis-acting regulatory consensus sequence on each of the genes in the group. Okay. So what is going to be there is going to be a specific transcription factor that will have a DNA binding domain and it will have a transcription activation domain. So the DNA binding domain will go and bind to the DNA, and the transcription activation domain, it will recruit coactivators such as histone acetyltransferases, which will make the histone which will make the histone less charged, and it will open up the DNA, and it will also recruit other general transcription factors. So this is known as combinatorial control. That is, a single specific transcription factor is recruiting a variety of proteins, and the specific effect of any one of them is dependent upon the entire protein composition of the complex. Okay. So basically what is happening, let's say there is a particular STF. Let's say there is STF3. Let's say there is STF3. It recruits GTF1. It recruits, let's say it recruits some histone acetyltransferase X, right? And it recruits some protein M. Okay. So the effect of STF3, let's say, will depend on this entire protein complex. So that is known as combinatorial control. So for example, if instead of protein M, it brings in protein S, then that will change the effect of the gene. So that is known as combinatorial control. The overall thing that is important for you to understand is that what is coordinate regulation, and what is combinatorial control. Although it's very unlikely to frame an MCQ from this particular point, but you will have to write this in your subjective paper. Just write these words at least. Okay? Even if you don't explain these, just write coordinate regulation and combinatorial control. It is done via specific transcription factors which have a DNA binding domain and a transcription activating domain. ठीक है, अभी ये थोड़ा मुश्किल लग रहा होगा part. Next we'll move on to regulatory elements. So there are proximal regulatory elements and there are distal regulatory elements. Distal means far away. Proximal means close to the coding sequence. So enhancer elements are located far away from the coding sequence and they will include variety of specific transcription factors which are responsible for giving a response to hormonal regulation. Then there are certain proximal regulatory elements such as CAAT box, CAT box, and there is a GC box that will respond to some specific transcription factor like CTF or SP1, okay? And they will regulate the frequency of initiation of transcription. So the distal regulatory elements will give response to hormonal regulation and proximal regulatory elements will respond to regulating frequency of initiation of transcription. Okay. So that's something that you can remember. Enhancer elements will help in response to hormonal regulation and they are distal. Okay. So now I will come back to an MCQ that I had shown you at the start of the class. Okay. Look at this MCQ now again. Which of the following are situated away from the coding region? So now your answer is very clear. The answer is enhancer because they are distal regulatory elements. They are distal regulatory elements which help in hormonal response. Much is clear to you? Why the answer here was distal regulatory elements was enhancer? Okay. So question number 2, the second question that I have for you is housekeeping genes are, this is a previous year question from JIPMER 2002. Again a very old question but very fundamental. What are housekeeping genes? Housekeeping genes are A, B, C, D. What's your answer? Guys, your answers? Okay. I think you guys are a little behind. There's a lag in the video. Guys, what's your answer for this MCQ? If you don't know the answer, please write I don't know. Please write I don't know so that I know that you guys don't know the answer and you want me to explain it. Okay, I guess we'll come back to our study and we'll revisit this question once you guys are alert. Okay. So we talked about coordinate regulation here, and I talked about regulatory elements. Next, I'm going to talk about the galactose circulate. Okay, so this is part of coordinate regulation. This is part of explaining coordinate regulation. In eukaryotes, we talked about the Lac Operon for lactose. Here we'll talk about galactose circuit. Galactose circuit is for utilization of galactose when glucose is not available. The Lac Operon was for utilization of for degradation of lactose when glucose was not available. Galactose circuit is also similar. It is to utilize galactose when glucose is not available. So I told you there is something that is equivalent to operon here, but it is not an operon. So that is known as galactose circuit, and it is part of coordinate regulation. It is part of coordinate regulation. Okay. So there is a specific transcription factor here. There is a specific transcription factor, that is STF, known as galactose 4 or GAL4. Alright, there is a GAL4 STF which binds to short regulatory DNA sequence of each of the genes for galactose. Alright, it is upstream to each of the genes which are required for galactose metabolism. Alright, therefore it is called as, these genes, they are called as upstream activating sequences, UAS GAL. Alright, UAS GAL. Look here. So let's say for galactose metabolism, you require 3 genes, A, B and C. Remember, in eukaryotes, genes are monocistronic. So these genes are present independently. Whereas in prokaryotes, genes which are part of the same metabolic pathway, they are generally present together. A, B, C would have been present together if it was a prokaryote. But since it is a eukaryote, all of these genes, they are not present on the same sequence. They are not present as part of the same unit, right? So therefore operons cannot exist here. That is the reason why operons cannot exist here. Instead of that, you have UASGAL, that is the upstream activating sequence for galactose, and that sequence is common for all of these genes, in upstream to all of these genes. And here you will have the binding, of a particular transcription factor known as GAL4, that is known as GAL4. It is a specific transcription factor. Okay, GAL4. So it has a DNA binding domain like all specific transcription factors, and with the help of that DNA binding domain, it will bind to this upstream activating sequence. It also has a transcription activating domain. It will have a transcription activating domain. Now the transcription activating domain, if it is active, it will invite other factors. Otherwise, it will be inactive. So what happens here is the binding through zinc fingers is in its DNA binding domain irrespective of galactose. So it will always remain bound to these short regulatory DNA sequences via its zinc finger DNA binding domains. Okay, that's the first thing. In the absence of galactose, there is something known as galactose AT, GALAT. It is also a factor, and it will bind to the GAL4 at its transcription activating domain. Thereby, it will inhibit transcription. So what did I just tell you? Look at the screen again. Look at my screen again. There was this gene that was required for galactose synthesis. Let's say it's called gene A. Okay, I'll do it with an actual gene. Let's do it with an actual gene. Let's say this is the gene for galactokinase. Let's say there is a gene for galactokinase. Okay, and there is an upstream activating sequence for this particular gene, UAS-GAL, right? On this gene, there is binding of GAL4. There is binding of GAL4 via zinc finger domains. So galactose 4, it is a specific transcription factor. So it has a DNA binding domain. DNA binding domain, and with the help of zinc fingers, it will remain bound here. Now, to activate transcription, its transcription activating domain must be active. Its transcription activating domain must be active. Let's say this is the transcription activating domain. Let's say this is the transcription activating domain TAD. But in the absence of galactose, what will happen is, if there is no galactose, then Gal80, there is another protein known as Gal80 that will go and bind here, and that will inhibit transcription from occurring. Okay, that's the first thing that you have to understand. But if galactose is present, in the medium, let's say galactose is present in the medium, then what happens? Galactose, it activates something known as Gal3. It will activate Gal3, and Gal3 goes and binds with, let's say Gal3 has this shape. Gal3 will bind with Gal80. And if Gal3 binds with Gal80, then this transcription activating domain, it becomes activated, and then it will allow transcription to occur. So now you understand how in the presence of galactose, coordinate regulation via the galactose circuit is possible. Understand? Gal4 is always attached here. But for transcription, for it to invite other transcription factors, okay, transcription activating domain has to be active, and for that Gal80 has to be removed from here. So in a way, Gal80 here, it is acting as a repressor, and Gal3 is acting as a inducer, if I am making an analogy to the Lac Operon. Okay. And if there is glucose, what does glucose do? Glucose, it inhibits Gal4. So glucose directly inhibits Gal4, and it prevents the utilization of galactose. So again, I ask you a question. Let's do it as a question. In which of the following states will Gal4 be active? Okay, if this is the question that is asked to you, and the options that are given to you are tell me, what is your answer? Very simple question. In which of the following states will Gal4 be active? Quickly, guys, I want an answer from you all. Again, if you don't know the answer, guys, simply say I don't know. Simply say I don't know. Okay. So it's very simple. Gal4, if it is active, it means that it should be bound to the DNA and it should be active. It should have a active transcription activating domain. For that to happen, there must be presence of galactose and absence of glucose. Presence of galactose and absence of glucose. So option B is the correct answer. So option B is going to be the correct answer for this particular question. So that is how you should be able to answer a question on galactose circuit. That is how you should be able to answer a question on galactose circuit. Okay. So you might be asked that when is gal3 active, when is gal80 active. Okay, so then you should be able to answer this. Gal4, gal80, gal3. Okay, that's how this entire regulation is. Here, if gal4 is bound with gal80, it will be inactive, and if there is presence of galactose, galactose will produce gal3, gal3 will bind gal80, and when gal80 would be bound, then gal4 will become active and gal4 will allow transcription of. So this is coordinate regulation. This is coordinate regulation via galactose. Coordinate regulation via galactose. Why is this called as coordinate regulation? Because multiple enzymes are being regulated by the single transcription factor gal4. Second type of coordinate regulation is called as hormone response elements, HRE. Okay, HRE. Hormone response system, HRE, hormone response elements. These are DNA sequences that bind trans-acting proteins and regulate gene expression in response to hormonal signals. Okay. DNA sequences that will bind trans-acting proteins and regulate gene expression in response to hormonal signals. Okay. So those are hormone response elements. Okay. So see here, there are certain intracellular receptors and there are some receptors that are cell surface receptors, right? So intracellular receptors, they themselves will function as specific transcription factors. But how are they different from other specific transcription factors? Is they not only have a DNA binding domain and a transcription activating domain, they also have a ligand binding domain. For example, glucocorticoid response element. These are true enhancers. So I will show you how what is going to happen. So let's say this is your glucocorticoid response element, which is an intracellular receptor. Okay, just like gal4, it is also a specific transcription factor. Okay. Why? Because first of all, it has a DNA binding domain which binds to DNA. Then it has a TAD, that is a transcription activation domain. Okay. Shabs Tasin, you are right. The answer was B to the question I asked previously. Then the third thing that it has is called as the ligand binding domain. It has a domain here known as the ligand binding domain. What binds here? Glucocorticoid or cortisol, right? So whenever a glucocorticoid binds here, the glucocorticoid response element is activated and it is bound to the DNA and it activates transcription activating domain, it attracts other transcription factors, and then that causes synthesis of genes that are present here. So it acts as a true enhancer. It acts as a true enhancer. So now you understand how a intracellular receptor, which is present generally for hormones which are lipid soluble, like cortisol is a lipid soluble hormone. So it is present for such hormones, and that is how it works as a true enhancer, and it is a hormone response element. It is a hormone response element. Okay. The second thing is for hormones which have cell surface receptors. Hormones which have cell surface receptors, one of the way they act is by increasing the levels of cyclic AMP. Then this cyclic AMP, it binds to a protein known as CREB. What is CREB? Cyclic AMP response element binding protein. It is a transacting factor which binds via leucine zipper. Okay, like you have zinc finger, you also have a leucine zipper, which acts to a cis-acting regulatory CAMP response element, which upregulates these enzymes. So what is happening here is, see here, this is your cell surface. Alright, this is your cell surface. Here you had a cell surface receptor. You had a cell surface. Let's say you received a hormone, and that hormone is hormone X, and that will activate adenylate cyclase. That will activate adenylate cyclase. What adenylate cyclase does is it converts AMP to cyclic AMP. Now cyclic AMP, it will go, let's say this is the CAMP molecule. It will bind to a protein. It will bind to a protein known as CREBP. It will bind to a protein known as CREBP. Okay, let's say this is the protein CREBP. That is CAMP binding protein, right? CMP response element binding protein. Okay, and then this entire complex, this is the trans activating factor. This is the trans factor. Then via the leucine zipper, it binds to, via a leucine zipper, it binds to a cis regulatory element. It binds to a cis regulatory element, which will further increase your. What is that cis regulatory element known as? We just saw that cis regulatory element is known as CAMP response element. Okay, or it can be known as CAMP CREBP response element. Okay, and that will upregulate certain enzymes. For example, your glucose 6-phosphatase is regulated in such a manner. Glucose 6-phosphatase is regulated in such a manner. Okay, glucose 6-phosphatase is part of your gluconeogenesis. It is part of your gluconeogenesis. So glucagon is responsible for increasing glucose levels, and that acts via increasing the levels of cyclic AMP. That acts via increasing the levels of cyclic AMP. So to simplify, let's say there was glucagon. Glucagon increased the CAMP levels. Then CAMP levels, or let's say instead of glucagon, I will take glucagon le lo. CAMP levels बढ़े, that will increase CREBP, and then that will increase the CAMP response element ka response, CAMP response element. That will cause an upregulation of enzymes such as glucose 6. Okay, that is how you will have to go about this particular regulation. So far what I have studied is, what we have studied, let's just summarize that. Regulation of gene expression in eukaryotes. We said at the level of transcription, in that we did two things. We did coordinate regulation via Galactose Circuit, and second we did hormone response elements. This is also example of coordinate regulation. Okay, this basic should be clear to you what you have to write. The second thing that we are moving to is post-transcriptional regulation. Second thing we are doing is called as post-transcriptional regulation, right? Should we move to this post-transcriptional regulation? You'll just excuse me for a moment. So I have a question for you meanwhile. This is the question. So tell me your answer, guys. Okay, guys. Back. Anyways, so this was a very simple question. I didn't get any answers from you guys. I think you guys are taking too much time. So the answer here is going to be option C, transcriptional regulation. This we just studied that galactose circuit is part of coordinate regulation, which is part of transcriptional regulation. Okay, so I'm going to start a different topic now, that is post-transcriptional regulation. So for people who have just joined, do pay attention and watch it live rather than watching it later, because then you can also ask your doubts side by side, right? So let's move on to post-transcriptional regulation. Let's move on to post-transcriptional regulation. So what are the things that you have to understand here is that there is something known as alternative mRNA splicing, then there is polyadenylation, then there is control of mRNA stability, and there is control of translational efficiency. There are 4 things that are related to post-transcriptional regulation. So remember, in eukaryotes, what happens is it does not end after transcription. Once you have transcribed DNA, it forms it does not form mRNA directly, it forms heterogeneous RNA. Then there is some processing that has to be done, like removal of introns, before you have the formation of mRNA. So there is some amount of processing involved. That is involved. So when post-transcriptional regulation is occurring, you are considering alternative mRNA splicing as the main mechanism. What happens here? Alternative mRNA splicing, it can be due to skipping of exons, it can be due to retention of certain introns, or it can be due to use of alternative splice donor or acceptor sites. Example, you have tropomyosin. Tropomyosin, it undergoes tissue specific splicing to yield a number of tropomyosin isoforms. And in over 90% of all genes, you have alternative mRNA splicing. Alternative mRNA splicing is believed to be one of the ways through which so for example, your body, how many genes does it have? Let's say it has around 1000 genes, and if you make it undergo alternative splicing, you can produce over 10,000 proteins just by the way of splicing it differently. So alternative mRNA splicing is responsible for the huge protein diversity, even though the number of functional genes are relatively less, whereas alternative mRNA splicing allows them to form varied protein products. Very different protein products can be formed from the same genes. For example, if this was one axon, this was axon number 2, this was axon number 3. Okay, in between them you have introns. Okay, so let's say one way to splice them was okay, to join 1 and 2, so that is one way to splice them. Other way to splice them was to join 1 and join 3. Okay, let's say the other way to splice them was one way to splice them removed the entire segment, one way removed this and this separately, and then you could have retained this intron, okay. So there are multiple possibilities in which splicing could have been done. So for example, this is one example of how alternative mRNA splicing will lead to formation of different products. Okay. So exon 1, exon 2, okay, that is the RNA that is produced. Alternative splicing, you can either have 1 2 3 4 5, you can have 1 2 4 5, you can have 1 2 3 5. So different varying proteins will be formed depending on which way you splice the protein products, which way you splice the mRNA will lead to alternative protein products depending on how it is undergoing splicing. So next I will move to alternative polyadenylation. So there are multiple sites for cleavage and polyadenylation, and it will generate mRNA with different 3' ends, okay, and that will also alter the untranslated regions or the coding translated sequence. That will alter both things. For example, the IgM, membrane bound IgM, is different from secreted form of IgM, and the difference between them lies in alternative polyadenylation. So alternative splicing, alternative polyadenylation, and alternative start of transcription site, this is one of the way in which using around 20,000 to 25,000 genes you are producing more than 1 lakh proteins. These are the various mechanisms responsible for production of this protein diversity from lesser amount of gene diversity. Okay, there are around 20 to 25,000 genes and you can produce more than 1 lakh proteins from them. That is because of these post-transcriptional modifications. They're very important. Okay. So next we'll move on to mRNA editing. This is a FAQ. This is frequently asked question. Okay. So Before I teach you this, I will ask you the question. Which of the following is responsible for formation of ApoB48? Okay, so I'll give you 4 options. First option is alternative splicing. Second option is alternative polyadenylation. Third option is mRNA editing. Fourth option is post-translational modification. Choose your answer quickly, very quickly. Choose your answer. If you guys don't know this, just please quickly write I don't know. Just quickly write I don't know. Very quickly write the answer I don't know if you really don't know it, because let's not waste time while we are going through these videos. Okay, especially for users who will watch it later, it will become very irritating if we waste time right now. Quick answers, guys, please. The correct answer is option C, mRNA editing. We'll just see how that is happening. So in mRNA editing, what happens is that's one of the bases they will get altered post-transcriptionally. One of the bases they will get altered post-transcriptionally. For example, in ApoB, in case of ApoB, the ApoB mRNA in case of liver and small intestine, it is ApoB mRNA is found in both liver and small intestine. But in intestine, what happens? In case of intestine, what happens? The cytosine base in case of this protein, there is a cytosine base that is present in the CAA codon, which codes for glutamate. But this cytosine, it gets deaminated enzymatically. Okay, otherwise its spontaneous deamination is also possible, but in the intestine you have an enzyme that will deaminate cytosine. So that will cause the conversion of cytosine to uracil, and what happens here? That forms UAA, and as you all know that UAA is a nonsense codon. That is, nonsense codon means it is a stop codon. And that will cause formation of ApoB48, and that is the one that is incorporated into chylomicron. Whereas ApoB100 is incorporated into VLDL, which is formed in liver. In small intestine, you have the formation of chylomicron. So in intestine, you will have enzymatic deamination of cytosine. This is known as mRNA editing, and this is not a mutation. This is being done by us specifically. We are doing this. This is not a mutation. Okay. And why is it called as ApoB48? Because it is only 48% of the complete peptide. It is getting interrupted in between. So for example, let's show you this in a proper way. If this was your, let's say this was CAA, and you had some protein after it. So this is 48% of the sequence. This is 52% of the sequence. And what happens if this is in the liver? In the liver, there is formation of ApoB100, and that is incorporated into VLDL. In the intestine, however, the cytosine base is deaminated to uracil. That leads to formation of UAA. That will be a nonsense codon, and that nonsense codon will cause premature termination of translation. That will lead to only 48% translation. So that will form ApoB48, which is a protein of chylomicron, which is this part of chylomicron. Okay, clear? What is mRNA editing? What is mRNA editing? So you have modified the mRNA. Okay. Cool. See. This is the same thing. It's simplified for you. Same flowchart. Cool. So next, let's talk about mRNA stability. Let's talk about mRNA stability. So you might have heard this in iron metabolism. You might have done this in iron metabolism. Okay. There are cis-acting elements known as cis-acting iron response elements, trans-acting iron response proteins. Okay. So try and understand this mechanism also. There are certain proteins known as IRPs. There are proteins known as IRPs. Okay. These are iron response. So when iron levels are high, these are all bound to iron. These are all bound to iron. Okay. These are all bound to iron. And when they are bound to iron, they are not present in the free form. They are not present in the free form. Okay. They are present in the bound form. But when iron levels will fall low, when iron levels they become low. So then iron response proteins they become free. These IRPs they become free. Okay, so you will have free IRPs. Now these will go and bind to iron response elements, which are DNA segments, which are nothing but cis-acting DNA segments. They will bind to iron response elements. Okay, they will go and bind here. What happens when iron levels are low in your body? IRPs will go and bind to IREs. That's how iron sensing is done. That's how iron sensing is being done. Now these IREs they can either be present on the 5' end or they can be present on the 3' end. Okay. So I will take this differently for transferrin and I will take this differently for ferritin. For transferrin, what happens is that this is the 5' end. This is the 5' end. This is the mRNA. This is the iron response element, and this is the 3' end. So when the iron response proteins they will bind here, they will stabilize the mRNA. And they will prevent its degradation. So they will help in increasing the synthesis of transferrin. If the iron response element is present on the 5' end, which is the case for ferritin. So when iron response proteins they will bind here, they will not allow the RNA polymerase to go over it. They will not allow the RNA polymerase to allow translation. So they will block translation. So in that case, they will block translation. Alright. So that's what you have to remember for ferritin and transferrin. And that is how they are controlling the stability of mRNA. That is how they are controlling the stability of mRNA via iron response elements. So when iron concentration is low, IRPs they will bind to the 3' iron response elements and stabilize transferrin. Similarly, opposite for ferritin. And amino levulinic acid synthase 2, it is regulated enzyme of heme synthesis and it also has 5' IRE, which means that if iron levels are low, ALA2 will not be translated, which means that you will reduce the heme synthesis. Why? Because iron levels are low in the body. You are not using it for heme synthesis. You are reducing heme synthesis. Okay. So three things are regulated by REs. What are the three things regulated by IREs? Transferrin, ferritin, and ALA2 synthase. Alright. ALA synthase 2. Okay. That is how IRPs will go and bind. They will increase translation if they are on the 3' end and they will block translation if they are on the 5' end. Okay, increase translation, block translation. This is true for ferritin and ALA2, and this one is true for transferrin. Okay, is this much clear? So let's summarize what we have done. Post-transcriptional regulation. Post-transcriptional regulation. What all have you studied? One, we have done alternative mRNA splicing. Then we have done alternative polyadenylation. Then we have done mRNA editing. Now we are doing mRNA stability. In mRNA stability, we are doing two things. One is what we have already done, that is iron response element. Second thing that we are going to do is called as RNA interference. You might have heard of this, RNA interference. You have studied this in your class 12th in your biotechnology. Okay, RNA interference. So what all are we going to do here? In RNA interference, what we are doing is let's see. It is mediated by microRNA. It is mediated by microRNA, and it is mediated by this microRNA is almost 22 nucleotides long. It is almost 22 nucleotides long, and there is something known as pre-mRNA, primary microRNA. On primary microRNA, you have an enzyme that acts, that is known as DROSHA, which is an endonuclease, and this will convert the primary miRNA into pre-miRNA. Then it goes from the nucleus to the cytoplasm, where you have an enzyme known as dicer, and this dicer will convert the pre-microRNA into microRNA. Once it becomes microRNA, there is a guide or the antisense strand of microRNA. It gets associated with the RISC protein. RISC is RNA induced silencing complex. RISC proteins. Okay, RNA induced silencing complex. They will get associated with the guide strand or the antisense strand of microRNA. They will bind to a complementary sequence in the 3' untranslated region of a target mRNA. They will bind to a complementary sequence in the 3' untranslated region of a target mRNA. That will lead to translational repression of target mRNA, and it can get degraded by an endonuclease. For example, the Argonaut or the AGO slicer of the RISC, right? So there are two different fates. One fate is translational repression, second fate is degradation by an endonuclease. Okay, and if there is an exogenous microRNA that is inserted, that is known as small interfering RNA. So what is the difference between microRNA and small interfering RNA? This is present naturally and this is artificial. This is artificial. Okay, so it is helping in inhibiting gene expression. So its role in the body is to inhibit gene expression, and its role outside is to we have its role in biotechnology. Exogenous miRNA, we have a role in biotechnology. Okay, so see here what was happening. There was formation of small interfering RNA which has an antisense or a guide strand which associates with RISC protein, that is RNA. See, RISC protein is RNA induced silencing complex, and then that RISC protein, it will go and either it can bypass translation, or that is stop the translation, or it can use its slicer. It can use its slicer to cleave the mRNA. It can use a slicer to cleave the mRNA. Okay. So we have the role of RNA interference based therapeutics, such as these have been used for the neurovascular form of age-related macular degeneration or wet AMD due to overproduction of vascular endothelial growth factor, and they're mostly in research phase. ठीक है, बहुत ज़्यादा अभी they are not used in humans. ठीक है, you might have read about the worms and plants in case of your class 12 है ना बायोटेक में. Now the next level of regulation is the translational level of regulation. Now what we did was the post-transcriptional. So we have done transcriptional, we have done transcriptional ka post-transcriptional. Now we are doing translational. Okay. The third level of regulation, translational level of regulation. Are you clear with everything else that we have done so far? Are you clear with everything else that we have done so far? We move ahead. Only some small part is left. After this, do you want to do another MCQ? Okay. You guys want to try question number 2 again? Housekeeping genes are. Okay. So housekeeping genes, I'll give you a hint, are the genes which are constitutively active. So since they are constitutively active, since these genes are constitutively active, it means that they are not regulated. So they are neither inducible, they are not required only when inducer is present, they are not mutant, they are the only thing that is there is that they are constitutively active. So there is no regulation for their gene expression. They are always expressed. Housekeeping genes are constitutively active because they are required for your cell process. For example, the genes for glycolysis. They are housekeeping genes. They are constitutively active. Alright. And other processes that are essential for the survival of the cell. They are all housekeeping genes. Okay. So first is the translational level of regulation. The first translational level of regulation, it will be done by the phosphorylation of eukaryotic initiation factor 2, EIF2, that is eukaryotic initiation factor of 2, that is a translational factor. It is a translational factor. You will phosphorylate it. And when there is phosphorylation of EIF2, it prevents GTP exchange. Alright, there is going to be a GTP exchange that was required for this step of translation, which was mediated by EIF2. And when you phosphorylate it, its function is inhibited, and that will inhibit the initiation step of translation. So first regulation, when you talk about translational level of regulation, is there is phosphorylation of EIF2, that is in eukaryotic initiation factor 2, that will prevent initiation of translation, okay. In this case what happens, how does the EIF2 get phosphorylated? Phosphorylation of EIF2, okay, it has to get phosphorylated. How will it get phosphorylated? By the help of kinases. And how are these kinases activated? These kinases, they are activated by some stress. For example, amino acid starvation, or deficiency of heme, or accumulation of certain misfolded proteins, or presence of double-stranded RNA. All of these are environmental stresses which will activate kinases. Kinases will cause phosphorylation of eukaryotic initiation factor 2, and that will prevent translation from occurring. That will inhibit the initiation of translation. Okay, this much is clear. First point regarding translational level of gene regulation. Second thing is that the regulation can also be done through variations in DNA. Okay. This is now a different kind of regulation. That we are doing. And this kind of regulation, it will occur when you control the accessibility of DNA. Just give me a minute. You'll have regulation through variations in DNA. So this is the next level of regulation. So far what I said was that there are 4 levels at which there can be regulation in eukaryotes. I said there are 4 levels. First we talked about transcriptional. Where we discussed coordinate regulation, Galactose circuit, hormone response elements, all of that. Then we talked about post-transcriptional regulation, where we said mRNA editing, alternative polydinylation, alternative splicing, right? All of that. Stability of mRNA, very important. Then we did translational regulation. That was phosphorylation of eukaryotic initiation factor 2. Then we said there is a variations in DNA. These are also responsible for regulation. Of gene expression. So you know that there are no histone proteins in case of prokaryotes, but in eukaryotes you have histone proteins. So they are very important for bringing about certain variations in DNA, and these are generally in the form of epigenetics. Epigenetics are something that you should know. They're very frequently asked, what is epigenetics? Okay, so it is gene expression which is independent of genes. Gene expression which is not dependent on genes but on other things like histone modifications, like histone modifications or chromatin remodeling. All of these are part of epigenetics. These are also inheritable. These are also inheritable, but they are not genes. Okay. So regulations through variations in DNA, accessibility of DNA, amount of DNA, arrangement of DNA, and transitions between various forms of DNA. So all of these are variations in DNA which can help regulate gene expression, So first we'll talk about accessibility of DNA to the transcriptional apparatus. We'll first talk about accessibility of DNA to the transcriptional apparatus. Okay. Since all of this is at the level of DNA, we are grouping this together. Access to DNA, first thing is presence of active chromatin. Okay. So histone proteins, they are modified at their amino terminal ends. Okay, they are modified at their amino terminal ends, and these modifications are known as epigenetic modifications. They can be of various types like methylation, acetylation, and phosphorylation. Okay. If there is a modification of histone protein in the form of any of these changes, what will happen? So at the amino terminal, there was a positive charge. Of the basic protein, because histones are basic proteins. There was a positive charge, but when you methylate this or acetylate it or phosphorylate this end, this positive charge is decreased. So there is a decrease in the positive charge of the basic protein. And since what happens? The histone is a positive charge and DNA is a negative charge. So when you remove this positive charge, this electrostatic interaction between them, it becomes weaker. Therefore, decreased association of histone with DNA. So histone becomes less tightly bound with the DNA, and that will relax the nucleosomes. So when the nucleosomes become relaxed, the access to DNA, it increases. Why? Because they are less tightly bound with the histones, and their availability or their access is much more. So that is the funda behind methylation, acetylation, or phosphorylation of DNA, and it is done by histone acetyl transferase. There is also deacetylation that will be done by histone deacetylase. Histone acetyl transferase, HAT. Okay, there is another HAT that is known as HAT medium that you study in biochemistry. What is HAT medium? Okay, one HAT is histone acetyl transferase. There is also a histone deacetylase which will remove the acetylation. There is another HAT in biochemistry, and that is known as HAT medium. What is HAT medium? You know what is HAT medium? It is used in case of monoclonal antibodies, I'll give you a hint. A here stands for aminopterin. H is hypoxanthine. Okay. Chalo. T is thymidine. You will study it later when we study monoclonal antibodies. For now, I am moving on to chromatin remodeling and Cpg islands. Okay. So we have done access to DNA via histone modification. Next we are doing chromatin remodeling. It is also known as nucleosome repositioning. And this is an ATP requiring process. So basically the nucleosomes are rearranged, and that is known as chromatin remodeling, and that is an ATP requiring process. The third thing that I am going to tell you is called as Cpg islands. What are Cpg islands? Why it is said Cpg islands? Why am I writing it like this? Because it is done in the Cg rich regions of promoter genes. In the CG rich regions of promoter genes, there is methylation of cytosine. There is methylation of cytosine. Cpg Islands. Cytosine, it gets methylated. Cytosine is the site where methylation takes place with the help of SAM, that is S-adenosyl. It is a methyl donor. It is a methyl donor. Okay. So cytosine gets methylated, and transcriptionally active genes are hypomethylated, but hypermethylation of DNA silences gene expressions. So CpG islands, they will silence gene expression. They will silence gene expression. How is that done? See here. So remember two things. If I will put methyl group on histone, if I will put methyl group on histone, that will lead to formation of more euchromatin, because it will decrease the positive charge on, so it will decrease the positive charge on histone, and that will increase the gene expression. But if I take cytosine and I methylate a nucleotide base, okay, that will lead to silencing of gene expression. That is lead to silencing of gene expression, because this is present in the promoter region. That will lead to silencing of gene expression. So hypermethylation of DNA is responsible for silencing of gene expression. Cpg Islands, very important, very important. So this is the part from where questions are generally asked. Access to DNA. This is very important for NEET PG. This is very important for NEET PG. Okay. Then the next point, this was the first thing that we discussed, was access to DNA. Now we will discuss amount and arrangement of DNA. Amount of DNA, for example, methotrexate resistance. Methotrexate is a inhibitor of dihydrofolate reductase, right? So what happens? In case of that, there is gene amplification. There is an increase in the number of dihydrofolate reductase genes, which allows thymidine triphosphate to be made. Okay, because DHFR is responsible for restoration of folate, and that folate is used to synthesize thymidine. Okay, that is responsible for this reaction. So it is very useful for synthesis of thymidine triphosphate. Okay, to convert uracil to thymidine. Folate is required, and that folate is restored with the help of dihydrofolate reductase. Methotrexate is an inhibitor of it. For methotrexate resistance, you will have gene amplification, which will increase the number of genes of enzyme. So amount of DNA is also responsible for regulation of gene expression. Last is arrangement of DNA. This you will study in immunology. What is Vdj recombination? It occurs in immunoglobulins. It is an example of somatic recombination. Alright. So Vdj recombination, it is a way of arrangement of DNA, and that helps in producing diversity in proteins. Okay, so different types of immunoglobulins can be produced because of Vdj recombination. To give you a hint, there are certain types of V chains, there are certain types of D chains, there are certain types of J chains. So let's say there are 20 of these, 30 of these, and 60 of these. So just for an example. So there can be any number of combinations that are possible between these three. Okay. So for example, V1, D2, J3 or V2, D5, J8. Okay. So ऐसे करके you can form multiple combinations. So arrangement of DNA will also determine what kind of protein is formed, and that is known as somatic recombination. Here it takes place in immunoglobulins. It's best given in QB's immunology. Alright, if you read QB's immunology, this is given in a very beautiful way, but not required right now for your understanding. Okay. It's somewhat like permutations and combination if you studied that in mathematics. Okay. B segment, D segment, and J segment. So there are multiple types, and they undergo recombination with the help of RAG1 and RAG2. These are enzymes that take part here. Okay. Then there are some pathological DNA rearrangements, which are, for example, translocation. Example, the translocation between 9 and 22. So there are certain translocations that are pathological, and they will also bring about regulation of gene expression. Last way to regulate gene expression is through mobile DNA elements known as transposons, transposons, which are approximately 50% of the human genome is believed to be in the form of transposons. And these transposons, they are moved with the help of an enzyme known as transposase, all right, which is coded by a gene known as TN gene. It can either cut, paste a segment, and it can either copy, paste a segment. It can either, so for example, if there was a transposon segment here, it can take it from here and paste it here, or it can replicate this segment and then it can take it somewhere else. So if it has to be replicated, then you have a RNA intermediate, and that is known as retrotransposon. And 90% of all transposons are retrotransposons. Okay, and there are some disorders in which, due to retrotransposons, you have hemophilia A or Duchenne's muscular dystrophy, right? They also play a role in antibiotic resistance. Okay, so transposons are one of the ways gene expression is regulated in eukaryotes. So gene expression in eukaryotes. Can I summarize this for you guys? There are 4 things that you will write mainly. One, you will write transcriptional. Second, you will write post-transcriptional. Third, you will write translational. Fourth, you will write DNA, variability in DNA. Okay. In transcriptional, what you will write, in post-transcriptional, what you will write, in translational, and in variability in DNA. Okay. So see here, first thing that you will write here, is there any other feedback? You will write here coordinate regulation. You will write here coordinate regulation. Then you will mention here galactose circuit. You will mention galactose circuit, and you will write about hormone response elements. You will write about hormone response elements. Okay, that much is done. In post-transcriptional, you will write alternative mRNA splicing. Then you will write about alternative polyadenylation. Then third thing that you will write will be mRNA editing. Fourth thing that you will write will be mRNA stability. In mRNA stability, you will write about iron response elements, and you will write about RNA interference. Then in translational modification, you will write about phosphorylation of eukaryotic initiation factor number 2. In variability in DNA, there is a lot to write. In variability in DNA, you will write here about, I'll tell you here. First, write accessibility of DNA. In accessibility of DNA, you have to write 2 or 3 things. First, you will write about histone modification or active chromatin, euchromatin and heterochromatinization. Second thing you will write here is Cpg Islands. Okay, and you can also write here about chromatin remodeling. You can write about chromatin remodeling. Then apart from accessibility, you will write amount of DNA. Then you will write about arrangement of DNA. You will write about arrangement of DNA. In that, you have to write about Vdj recombination. You can also write about translocations. Then your last point is going to be mobile DNA elements, that is transposons. So this is a summary slide of what your ideal answer should be, and what should all the points be in your head when you are considering regulation of gene expression in eukaryotes. I've already discussed the summary slide for regulation of gene expression in prokaryotes. This is the summary slide for regulation of gene expression in eukaryotes. I hope it is clear in your head now what you have to exactly write, and the concept map is clear in your head regarding how you have to present everything. Okay. Is it clear to you? Any doubts? Doubts, if any, please tell me quickly. I'm going to move on to MCQs now. If you guys liked the video so far, you can hit the like button. I would really appreciate it. And do subscribe to the channel as well. And you can share this video with all of your friends who are preparing for their prof examinations. This will definitely be useful to them. And if you take an Unacademy subscription, do use my referral code Chetanya10. Any doubts that you guys have? I will only move ahead if you guys say that you don't have any doubts. Please someone write in the chat box that you don't have any doubts. Okay. So let's move on to question number 3. Which of the following is true about transposons? Which of the following is true about transposons? So we just studied what are transposons. And let me remind you, this is a question from the PGI examination. So this is a multi-answer correct. This is a multi-answer correct. So I'm looking for your answers in the chat box. This is a multi-answer correct. So I'm looking for your answers in the chat box. Okay. Deepika is here, and Deepika is saying that the answer is only option B, also called as jumping genes. So that's correct, that's true, but there are other options also that are correct. Like I told you, this is a multi-answer correct question. So there are multiple answers which are correct, not just one. So you have to choose other options here also. Okay. So choose between A, C, D. Okay. Other options. This is a multi-answer correct question. So now Deepika is saying D is also correct. It is called as retrotransposon when it involves RNA intermediates. This is again true. This is again very true. But there are other statements which are also true. There are other statements which are also true. Okay, one more answer. Come on. So the thing about this question is that A, B, C, D, all four options are correct. I was hoping to get this response very accurately from someone. Uh, it is sorry, A is not correct. It has no effects on gene expression. This is false. B, C, D are correct. It is also called as jumping genes. It is mediated by the enzyme transposase, and it is also called as retrotransposon. It does have effects on gene expression. It is part of regulation of gene expression in eukaryotes. So B, C, D, these three answers are correct for this particular question. Not all, Shabstasi. A is incorrect. It has effects on gene expression. Okay. B, C, and D. These three are correct. Okay. So that is question number 3 that was asked in PGI in the November of 2013. So question number 4 again from the same shift. So you can see in the same entrance examination, this question has been asked from same topic, that is regulation of gene expression. So it is a very high yield topic. ठीक है, कभी कभी बिल्कुल भी नहीं पूछते, बट कभी कभी जब पूछते हैं तो दो दो क्वेश्चन पूछ लेते हैं. Alright. So genes in the CpG islands are inactivated by. तो आई टोल्ड यू अबाउट सीपीजी आइलैंड्स राइट नाउ. ठीक है, तो बहुत स्पेसिफिक क्वेश्चन है, देखो. Question number 4, what is your answer, guys? Okay. Chubb's Tassin is saying A, and that's absolutely correct because methylation of cytosine residues here, methylation of cytosine residues here, that will lead to inactivation. That will lead to inactivation of gene expression, silencing of gene expression. Very correct, very correct. So next we'll move on to question number 5. We'll move on to question number 5. Inactivation of X chromosome is called as. Inactivation of X chromosome is called as. Okay, so Deepika has given the right answer here. This is known as Lyon's hypothesis or bar body inactivation. Lyon's hypothesis. That is called as bar body inactivation. Bar body inactivation. So from the name of the scientist, it is derived Lyon. Okay, that is known as lionization. Okay, plays a role in the regulation of G expression also. Okay, question number 6. Question number 6. This was asked in AIMS in 2011. No, the answer is A here. Allelic exclusion is different. It will come in immunology. Allelic exclusion, this will come in immunology. Okay. Guys, answers. Histone acetylation. It's very logical. Histones are basic proteins, so they have positive charge. So what will happen if you add acetyl groups to them? What will happen if you add acetyl groups here? You will neutralize this charge. What will that cause? What will that cause if you remove this charge? No, Deepika, your answer is wrong. It's not DNA replication. Histone acetylation, if there is histone acetylation, let's say this is a histone protein. It is a basic protein, so it has this positive charge and it binds to negatively charged DNA. So if you acetylate it, this positive charge is reduced. So that will decrease its association with DNA. Decreases its association with DNA. And if the association with DNA is decreased, that will lead to increased euchromatin formation. What is euchromatin? That is, the DNA is less tightly bound. DNA is less tightly bound and it is more expressed. It is more easily expressed. So the answer for this question is going to be option B, increased euchromatin formation. Increased euchromatin formation. Okay. Next, question number 7. Pyrimidine dimers are seen in which of these scenarios? Question number 7. Okay. The correct answer here will be xeroderma pigmentosa. That is where you have errors in repair of pyrimidine dimers. So you will have xeroderma pigmentosa, right? Question number 8. Which of the following is true about DNA methylation? Which of the following is true about DNA methylation? This is a question of PGI 2010 November. And which of the following is true about DNA methylation? So like it is a PGI question, so I keep reminding you this is a multi-answer correct question. You have to choose multiple answers. Which of the following is true about DNA methylation? Okay, it is a multi-answer correct, Deepika. So protective mechanism against cleavage by restriction endonuclease. This is absolutely true. D is correct. You are right. But there are other options also which are correct in this case. DNA methylation is an epigenetic modification. It is an epigenetic modification. Please do remember that. Since it is an epigenetic modification, genetic code, it remains intact. Genetic code is your DNA sequence. So that remains intact after DNA methylation. So that's right. Deepika is right. B is also correct. There are more options that are correct in this case. There are more options that are correct in this case. It brings about alteration of gene expression. That's absolutely true. DNA methylation brings about alteration of gene expression. Correct. A is correct. Very good. Now what is left? C. C is also correct. It also has a role in carcinogenesis. DNA methylation also has a role in carcinogenesis since it is responsible for silencing of genes, since it causes silencing of genes. So if a tumor suppressor genes get silenced, that will lead to carcinogenesis. So that is one of the mechanisms in which you can have a role in carcinogenesis. Okay. So question number 8, all the 4 statements are true. Question number 9. Question number 9, this was asked in AIMS 2014 May. So like I told you, the portion of eukaryote regulation, especially the one related to DNA modifications is very important. DNA modifications is very important because it is also clinically relevant. The part related to DNA modifications. Yes, it can be carcinogenesis also. Right. I think this is the last question for today. Question number 9. So the answer is going to be very simple. Methylation of cytidine residues. So think about Cpg islands. Think about Cpg islands. It is in the promoter region. These are present in the promoter regions, which are Cg rich. Right. So Shabs Tasin has given the correct answer. There is a decrease in gene expression. Hypermethylation causes hypermethylation of DNA causes a decrease in gene expression. Okay. So that's the end of today's class. If you liked the class, do remember to hit the like button, and once the class is over, do drop a comment in the comment sections saying that you liked the class. Do hit the like button and also subscribe to the channel Let's Crack NEET PG. You can also follow us on Telegram, right? You will be updated with the classes. My Telegram channel is @Unacademy, NEET PG Chetanya, and the Telegram channel for the YouTube channel is the same as the name of the YouTube channel, that is Let's Crack NEET PG. And this is my plus referral code. This is the plus referral code I would encourage you to use for taking any plus or iconic subscription. You'd get a straight away 10% discount. Okay, you can also use the referral code SPCM. And if you haven't downloaded the Unacademy learning app, please download it and use this referral code to take a free plan. If any of your friends or relatives or anyone you know is taking a plus subscription, do encourage them to use my plus referral code. Hit the bell icon for further notifications. So I'll put the link of the Telegram channel here in the chat. And you will also find in the description, of this video, you'll find the link to my Unacademy profile, right? So shall we? Okay. Thank you, Shops. Thank you, Deepika. Thank you for watching. Okay. You can drop a comment once I've ended the stream. You'll have the option to drop a comment so that will stay. This is a live chat. So once the live chat is over, once the comment is streamed, so I would really appreciate it if you could drop a comment. Thank you so much.